I'm going to be talking to you about is our work in our lab trying to figure out how to improve and manage cover crops, especially legume management in organic cropping systems to try to improve soil fertility and health. And some of the work I'm going to present today is carried out by these individuals here. Sarah Seehaver, who's my lab technician, Mary Parr, Matthew Matapo, and Matthew Brown, who those three are graduate students in my group. So today, first thing we're going to do is just go over some definitions. I know all of you know what a cover crop is, or you probably wouldn't be in here, but there's some fuzzy zones that I want to clarify so that the presentation is clear. And then we're going to talk a little bit about soil biology and try to walk you through the key process that when you're using legumes you need to understand in order to manage them effectively and efficiently. I'm going to talk to you then about some of our data, showing some of the data from spring termination of cover crops, when we do it, how we might manage that process better, and finally talk about how much nitrogen we're going to get from our legumes and when we get it. So in 1990, there was the Organic Food Production Act was passed by Congress, and it was enacted a few years later and put into place where now all but the smallest organic growers were required to adhere to certain practices in the U.S., requiring them to farm in a certain way, of course. So when we think about soils, there are a few parts of the code that are important to understand, and you'll see as I go through it, they can be somewhat vague. So we've got some flexibility within how we manage, but these are overall goals in organic agriculture that we want to achieve. So they all have these funny little numbers here. 205, 203 is the soils code. We need to select tillage that maintains or improves physical, chemical, biological condition of the soil. So in effect, what that means is we want to move towards reducing tillage in organic systems. That's going to become important later when you see I describe some of the work we're doing. We want to include rotations, cover crops, and plant animal materials as our primary soil fertility source. So these are organic residues. These are complex carbon-based residues that are applied to increase fertility. We want to actually also maintain or improve soil organic matter content. You notice it says maintain. We don't want to do anything that's going to reduce soil organic matter. We essentially want to try to increase it, but times we can't. We want to just improve it. So we want to do this through the application of certain techniques. Let's see if I can do this. That are going to not contaminate soils. And you think about organic, how are we going to contaminate soils? Well, if you apply too much of one type of material or another, you can have contamination, especially in the case of organic agriculture, through application of manures that might increase phosphorus loading, et cetera. So we want to be aware of that and try to limit the amount of nutrients we're putting on of any one type. Another part of the code, 205.205, is the crop rotation standard, and this states that a producer must implement a crop rotation including but not limited to sod, cover crops, green manure crops, and cash crops. So you can see all of these rules and regulations are sort of steering us towards the use of cover crops in organic systems to improve fertility. When I talk about a cover crop, I use it in a very general way as sort of an umbrella term to talk about a crop that's planted between periods of regular crop production that provide benefits other than food production. And in many cases, those are soil fertility and soil organic matter improvement. However, cover crops, of course, are also used for a number of other goals, to achieve a number of other goals as well. If you look at this little triangle here, some of the characteristics on the top, adding organic matter, suppressing weeds, adding nitrogen if it's a legume, are some of the more popular reasons people are using cover crops. And then you have these other goals at the bottom here, improving soil structure, suppressing nematodes, increasing water infiltration, attracting beneficial insects. These are other benefits, and many crops don't have all data. You can't do all of that in one silver bullet cover crop, but you can prioritize the type of crop you're going to choose so that you can achieve a few of these at any one time. So here's the definition part. You've got this general umbrella term of cover crop. Some people, when they refer to a cover crop, means a crop that is simply in place to reduce soil fertility or reduce erosion and soil loss. But then you also hear this term green manures. Green manures are legume cover crops. So those are cover crops used specifically to improve soil fertility through this nitrogen-fixing symbiosis, which I'm going to describe in a second. And then you have catch crops, which are also another type of cover crop. Can be a catch crop as well. The cover crop is used primarily to retrieve available nutrients that still might be following, say, manure application or other materials in a field that you don't want to be building up in your soil over time. So they're used somewhat interchangeably. So you want to be aware of these terms and how they're used, and I probably will use them interchangeably throughout this talk as well. I love going out to my fields in the spring because when I go out there, this is what I see. I see this beautiful patchwork of different cover crops, and in my case, we're looking at legume cover crops and flower. So some of the more popular ones in the state include crimson clover, hairy vetch, and Austrian winter pea. And those are the three I'm going to be talking about when I present our data today. So when thinking about planting a cover crop, we have some time constraints and some windows that we need to think about when we're planting. So when we're thinking about planting a winter cover crop, we're going to plant it in the fall. It grows a little bit through the winter or until it gets really cold, and it's dormant in the winter. Grows a lot in the spring, and then we kill it sometime before cash crop planting in the summer. And that's, in particular, we're working with corn. But if you're thinking about other planting windows for cover crops and other crops, other cash crops that you're going to be planting following cover crops, you have this window of time between September and November when you could plant them. And you can see that in some cases it doesn't fit in very nicely with some crops like soybeans, cotton harvest. So in these cases, you may want to think about aerial seeding, or as Francis Tick was talking about yesterday, cross seeding is also common in some places in the north. I'm not sure how well it would work down here. But where it fits in nicely is after corn. But we're still having some challenges with that, as we'll describe in a little bit. So the key process that I feel that growers should understand in order to manage effectively their legumes, cover crops, is this process of nitrogen fixation. And what this is a symbiosis or a relationship between a plant, a legume plant, and soil bacteria, where the soil bacteria is basically grabbing nitrogen from the atmosphere, fixing it into a form that plants can use. Because how much of the atmosphere is nitrogen? Who knows? About 80, yeah, so 70, 79. And taking that nitrogen, fixing it into a form the plant can use. The legume uses it, it ends up in the biomass of the plant, and then when the plant dies, it makes that material, that nitrogen, available to non-legume plants. So that's great and grand. The legume is getting nitrogen, but what is the bacteria? Does anybody know what the bacteria is getting from that relationship? Living in the plant roots, soil bacteria. Sugar, yeah, it gets sugar from the plant. So let's walk through this process. You've got your legume plant growing in the soil. If there's no rhizobia, it's not going to have nodules. It's not going to fix any nitrogen. If there's rhizobia in the soil, it'll form nodules on the plant roots. It will fix the nitrogen. That plant will green up if it's fixing a lot of nitrogen, because all that nitrogen from the atmosphere ends up in the leaves. And the reason I want to focus on this, because I know a lot of you have seen this before, is because this process of the nitrogen being fixed and carried up into the leaves, that's the key point of this. A lot of people think that the nitrogen is actually leaking out through the nodules. That's not what's happening. The nitrogen is being fixed, carried up into the leaves, and then when that plant dies or is killed through spring termination, that nitrogen then becomes available in the soil for other plants to use. So then when you plant, say, your corn, the corn can then take up that nitrogen that's released into the soil. So that's the whole process. There are two places at which you can manage this process. And so our lab as a whole tries to emphasize both of these and trying to learn how we can effectively manage legume cover crops in organic systems. Number one is managing the symbiosis, managing the bacteria that are living inside of those nodules. Number two, we want to manage the residue. We want to manage the ways in which we can improve the amount of material, the nitrogen that's being put into those leaves, improve the amount of biomass the plant's putting on, thereby getting more nitrogen from it, and the ways in which we are terminating or killing that cover crop in the spring so that that nitrogen becomes readily available for the cash crop following the legume. Now, there are a few things that you should know about nitrogen fixation in general that we well understand. One is that nitrogen fixation is very, very expensive for the plant, meaning that it spends a lot of its time giving carbon to these root nodule bacteria. So if there's a little bit of nitrogen in the soil, it's going to form more nodules and fix more in. It's going to fix more atmospheric nitrogen. You see how fat that arrow is. However, If there's a lot of nitrogen in the soil, available nitrogen, that plant's going to say, well, I don't need to form nodules. I don't need to fix nitrogen. I'm going to take more out of the soil and fix less. So that little arrow is somewhat small. As soil, if you're working with a very fertile soil, so say organic farms, some of the organic farms we worked with in New York State had been applying compost and manures and other organic amendments for 20 years. They had very high soil N levels, and we found that nitrogen fixation levels on those farms were low because the legumes were taking up nitrogen from the soil and not using it from the atmosphere. Second thing you want to keep in mind is that the bacteria population in your soil of Rhizobia that are hanging out, ready to fix nitrogen, or ready to form nodules, can change based on your management. In this case, we looked at organic farms across North Carolina where we had paired fields, some fields where they had been using, in this case, hairy vetch, cover crop, leaf cover crop, for years and years, and some where had never, in the same farm, they'd never used hairy vetch. And just by nature of them planting hairy vetch, these farmers had increased their levels of Rhizobia in the soil so that when we planted a legume into it without adding any more bacteria, you get nice, healthy plants. Where the field that had never had hairy vetch planted to it, we often saw no nodulation, and I don't know if you can see that, like very dead plants. This came out in the data too. If we look at three different farms where we have hairy vetch, history of hairy vetch planting in the red, and the yellow were farms where they had never planted hairy vetch. We saw across the board lower nodulation in fields that didn't have history of hairy vetch. So just planting the legume, even if you're not adding an inoculant, it's always good to add an inoculant, but even if you're not, can change that population. So thinking about whether you want to add an inoculant to a field or not is pretty important. If you have a field that is new, like the fields I was pointing to in the previous slide, that had never had hairy vetch, had never had a legume planted, you probably will want to inoculate. And the bacteria and the plant relationship is very specific. So when you're buying a new legume, a new type of legume for a field, you've never planted it, you want to figure out what that group of Rhizobia is that will form nodules on the plant. And then at planting time, add that bacteria to the seed, plant the seed, and then that bacteria will be present when that seedling starts to form roots. These, I know you probably can't read this, but these are some of the common groupings of Rhizobia. Here being alfalfa, common beans, clovers, cowpeas, and then the vetch and pea. So there are some plants use the same type of Rhizobia. For example, Austrian winter pea, lentil, and vetch all use the same Rhizobia type. So if you go into your farm store and you ask for your cover crop Rhizobia that nodulates this particular plant, it might sometimes be the same type. Mm-hmm.
I've heard that you said one about active inoculants that years ago, I think someone up here in North Carolina told me that the seeds go buy an inoculant, it's probably dead.
Yeah, so the question was quality of inoculant and whether you are, if you just go to a seed store and you're buying an inoculant straight off the shelf, whether it's going to be alive or dead. That's a very good point. Inoculants are live bacteria, and so you need to have certain precautions that you take on your farm. Leave it on the dashboard of your truck, yeah, you're not going to have very much that's alive in that truck after a summer in North Carolina. It should be refrigerated, and you should ideally only keep it for a year after purchase. And we don't, the honest truth is, you don't know what's happened to it when you're buying it from the seed store prior to you purchasing it. You might treat it great, but when you get it, there's no way to tell how it's been treated prior to you purchasing it. So we really don't know, and we actually don't know how growers are working with inoculants after they're purchasing it either. We actually did a survey last year, and we haven't analyzed the data for it yet, but trying to figure out how farmers are managing or using their inoculant, both storing it, applying it, especially with organic producers, or using all sorts of things to make it stick to the seed. Some are using milk, some are using sugar water. So we want to get a sense of what growers are using across the state to manage that inoculant. That's a very good question. It might be dead, but it's a precaution that you should take anyway, even though you don't know what's happened before you purchased it. Applying it to the seed is a good thing to do if you know that field has never had the bacteria in it. So one of the other things that we've looked at in the past is this history question of planting the legumes in the field, what that does to not only the number of bacteria, the number of Rhizobia that are in the soils, but the diversity. See if we can create a range of bacteria that live in those soils able to nodulate new legumes that we might introduce. So in this study that we did up in New York State, we've compared organic farms to conventional farms. The organic farms were farms that had often used multiple different types of crops, multiple different types of legume cover crops in addition to their cash crops. And we found that organic fields in general had a more diverse array of Rhizobia. Some of the reasons this might have, or these fields actually had less synthetic inputs or no synthetic inputs that they'd used in the case of the organic fields. And they tended to have this diversity of crops in the field history. And when we compared those to farms that they're conventional farms that had just a monoculture or very reduced diversity of crops in the background, we found a much reduced diversity of Rhizobia. So there's a lot of evidence out there. We're starting to gather more evidence that the cropping system that we choose and the diversity of crops that we're using is going to impact the Rhizobia that are already there in the field. And this becomes important to us because when we think about inoculation, it could be that later on down the road, after we planted a legume for a number of years, that we might not need to inoculate every year, but we don't know that yet. So until then, we need to just take precautions and inoculate our seeds in the field. Okay, so we've talked about a little bit about managing the bacteria. Let's talk about managing the residue. When you are killing your cover crop in the spring, many farmers would mow it and then disc it in. So this is more of the common standard practice of killing your cover crop and making that nitrogen available to a subsequent crop. But of course, we talked about earlier the goal of wanting to reduce tillage. So tillage in cultivation is used in organic systems to try to reduce weeds, but it's also used in the case of cover crops to kill that cover crop. So how can we kill the cover crop without using lots of tillage? Well, one of the things we've been playing with is this. What's this? Roller. Yeah, how many people either have one or have seen one of these in action? Anybody in here? Yeah. So we've been trying to make this work with legume cover crops, and what happens is this water-filled heavy drum is pulled either behind or pushed in front of a tractor, and then the cash crop is planted right into that mulch. So the mulch theoretically produces an effective weed control barrier as well as providing nitrogen or another nutrients as it decomposes to the crop that's growing in it. Let's see, it should be a movie. There we go. So this is what it looks like. It's a little choppy, but this is what it looks like in action. This is Austrian winter pea, and that's what it looks like afterwards. It always looks great afterwards, right afterwards, and sometimes it'll come back a few hours later, and it's all popped back up because the plant is hardy enough. If it's still actively growing, it's hardy enough that roller isn't going to kill it. So what we found we need to do is manage it so that we're killing it at very, very late in its life cycle. It differs by species, and I'll show you a little bit of the data in a second. But by killing it later in its life cycle, that plant has channeled and is spending a lot of its energy making seeds, forming flowers. So you can effectively kill it later in the season if it's very close to senescence. Oops, there we go. So I mentioned weed control. Weed control can be excellent in rolled kill systems. Here you see some of Christopher Horton's work looking at rolled rye and soybean. So from our fields, using rolled crimson clover with corn, and you see one big weed there, but for the most part, it's looking pretty good. The problem is, oh yes, go ahead.
When you roll it, leave it on the top, and don't incorporate it, when does that nitrogen from that crop become available?
Yeah, the question was if you leave the mulch on the top, when does that nitrogen become available? And I'm going to present some preliminary data on that. We're trying to figure that out right now. It's a very good question. So one of the considerations you need to take when you're thinking about these roll systems is because you need to kill the cover crop later in the season, you have a problem with then not being able to get the corn in the ground until later in the season. So I don't know if you can see this, it's a little late in here, but here you see in the left-hand side the corn that was planted in mid-April. Cover crop killed, corn planted the same day, mid-April. Planted early May, planted mid-May. So the mid-May and early May look pretty good, but that's pretty late for corn. Planted mid-April, the problem with this is that a lot of the cover crop was still alive. It was competing with the corn seedlings that we planted into it, and you end up having lower yields from that corn. Here's an extreme case when we planted the corn very early at the same time early for the cover crop, mid-April, you end up having a lot of competition with the corn from that live cover crop and not much growth. So let's look now at the amount of nitrogen that's contributed. So this has to do with the question that was just asked about from David about when do we get the nitrogen and how much nitrogen do we get? And this is really what we're most interested in. We want that nitrogen to be the most possible, that plant to be fixing the most possible nitrogen it can and delivering it to the corn upon kill during the decomposition process. So what you're seeing here is legume biomass. This is in kilograms per hectare. I'll try to do some translation as we go through it to pounds per acre. And these are cover crop varieties, species and varieties. So we have varieties of hairy vetch, some different crimson clover varieties, and then some others which I'm not going to mention much about today, and Austrian winter peas. Those are the three that we've really taken an interest in here in North Carolina. What we found, by and large, is that hairy vetch was really the winner. It came out on top in terms of the amount of nitrogen that it could produce about 150 to 180 pounds of nitrogen per acre, followed by crimson clover and Austrian winter pea. All of this biomass data, however, you think, wow, this is great, we got a lot of nitrogen, but all of this data is from mid-May. So this was the most ideal time to kill that legume cover crop effectively. And of course, because it got more time to grow, we ended up having more nitrogen in the cover crop that then could be made available to the following corn. Another thing you want to think about when we're thinking about the legume, the amount of nitrogen contained in the legume, is that because it relies upon that microbial process of decomposition to make it available to the cash crop, not all of that 180 pounds of nitrogen is going to be made available to the cash crop right away. It's slowly decomposed over time. So it's going to, if you have a roll system, it's sitting there on the surface, and it's going to be slow to decompose. Yeah.
I was wondering if you would slide here in days, how soon after you will come crop field will you come back?
So you're asking how soon after you do a cover crop kill can you come back and plant? You can do it. We do it immediately. We do it the day after or the day after. So often we'll wait a day to make sure it's killed and then plant into it. But yeah, it's almost immediate. Some people actually put the implements, the planter on one side of the tractor and roller on the other, so roll it and plant it. So you can play with that a little bit, but usually it's right away. So here, yeah, that nitrogen that's in that legume it won't be made available right away. It's going to take time for that to decompose. So let's break that up a bit, looking at those different plant dates. What you just saw was data looking at the best, the optimal date for killing the cover crop using the roller and how much nitrogen we get on that optimal date. Let's look at the other dates. Here we're looking at hairy vetch, and these are the different varieties of hairy vetch that we were evaluating. This one here is an interesting case. This was USDA variety, Full Prosperity, which was supposed to flower earlier. So by flowering earlier, it means that we can kill that cover crop a little bit earlier in the season and get our corn in earlier. So here we have the amount of nitrogen produced, and as you can see, in all cases, the nitrogen went up the longer we waited, which seems logical, with the most nitrogen in that plant being achieved at that late roll date of mid-May.
And that's nitrogen available in the crop.
Not a bit, yeah, it's the nitrogen in the crop.
Right.
Exactly. So then it's going to need to go through that decomposition process to become available to the plant. So the question was that the data I just showed, if that's the nitrogen in the plant, not the nitrogen that's available to the crop. Yes. So I'm repeating for the webinar audience. Yes.
The seed viability of the early flowering vetch, problem when you're waiting for this international level to come up, and then
so your question is what seed viability in the vetch?
That early flowering vetch, which you want ideally for your crop,
yeah.
End up being a weed problem because that seed is more available for a bit more viable.
Okay, so yeah, the question is whether that seed of the vetch that's early flowering will actually be viable enough to become a competitive problem. And no, you should not be letting it go to the point where the seed is viable. You're catching it, when it's maybe 3/4 of the way flowered across your field, maybe forming very thin pods, not filled out pods, and then you can effectively kill that cover crop. Yeah, that's a good question. There are cases though with crimson clover that we did have viable seed when we were looking at these different kill dates. So here are the same kill dates you saw before, different varieties of crimson clover. And in this case, we saw that the most nitrogen, when we killed it at the mid date in late April, that's when we had the most nitrogen. And you wonder, well, how could it have lost nitrogen later in the season? Shouldn't it just be getting bigger? Well, this was because in some of these cases, we think the nitrogen, if they had formed viable seed, the seeds had actually shattered, and so we ended up losing some of that nitrogen and having it kind of depleted leaf matter, depleted in nitrogen. And so for crimson clover, we find that it actually has the most nitrogen at an earlier date. So that might be a more reasonable time to get your corn in the ground at those mid dates. At some of these late dates for crimson clover compared to hairy vetch, you can actually see the difference in the corn. Can you see how yellow that crimson clover mulch is making the corn look? It's likely depleted in nitrogen because how many people were in Francis Tick's talk yesterday? Right here, him talk about mobilization, nitrogen. That's what we think is happening here. That when you have hairy vetch that's too old, it's too stocky, too woody, very carbon rich, the microbes are trying to decompose that mulch on the ground, and there's not enough nitrogen in it because the seeds have taken most of it away. There's not enough nitrogen in that mulch for the microbes to do their thing, so they're sucking the nitrogen out of the soil in order to break down that very high carbon mulch because we let that hairy vetch go, or that crimson clover grow so late. The hairy vetch mulch-produced corn looked good at that late roll date. So it's going to depend a little bit on when you kill that crop. So for you guys as growers, how much nitrogen is in your cover crop? How do you figure this out? There's some rules of thumb that you can use when you're thinking about legumes. So of course things like hairy vetch, crimson clover, Austrian winter pea. If before flowering or right at flowering, that's when it's at peak nitrogen fixation, and this is, hasn't sent a lot of that nitrogen to the seeds yet. You get about 3 to 4%, and if it's a little bit after flowering, it can be reduced to 2 to 3. Usually we see around 3, so it's not that low. And non-legumes, 2 to 3%, 1.5% at flowering, and other non-legumes, same as grasses. So if you want to figure this out for yourself, you can. You can utilize the NCDA plant tissue testing labs to do this. It's a great resource, and I encourage people to do it. You can take a frame of known dimensions into your cover crop field, throw that frame down, cut down all biomass of the cover crop standing within that known dimension frame, send it away to NCDA, figure out the percent nitrogen you have in that cover crop, multiply it by the amount of stuff you have, and you can get the amount of biomass or nitrogen that you have on an acre basis for your farm. And sometimes I walk growers through that process in workshops. If any of you have ever been in a workshop with me before, it's a very easy thing for growers to do. And to get your sample analyzed for exact nitrogen, so you don't want to use those rules of thumb I just presented to you, you can send it to NCDA, and it's about $5 a sample for North Carolina growers. If you're out of state, it's $25 a sample. It's a great deal for those of you living in North Carolina, so it's something you definitely want to consider. That's plant testing. Soil testing is free. I mentioned that yesterday to a group, but that's an important thing. I think we're the only state left in the country that still has free soil testing.
Is that the plant tissue analysis?
This is plant tissue that I'm showing here. But this, yeah, so you can send this away. There's instructions on how to fill out the form, where to send it, what they need to know on their website. This is their website. But I just wanted to mention that soil testing is free. This is $5. So the last thing I want to talk about today is timing of nitrogen mineralization. This is related to David's question, where typically if we incorporate the cover crop upon termination, that mineralization process, that release of nitrogen in that material takes place earlier than when the crop needs it. So we want to figure out how we can better time that release of nitrogen so that crops can get it. We've been playing around a little bit with looking at the release of nitrogen from cover crop legumes that are terminated or killed in different ways in the spring. So we're comparing these the more traditional method of disking them in. This is brand new data, so it's a little rough on the slide here. But we have the amount of nitrogen when it's disked in, the amount of nitrogen when it's just flail mode, it's sitting on the surface, how much is in the amount of nitrogen released when it's rolled, and the amount of nitrogen when it's sprayed. It's more of a conventional method. And this data right here is for Austrian winter feed. The yellow bar up top is showing time. This is time after cover crop kill. These are plots that have been fertilized with synthetic nitrogen, about 150 pounds per acre. Down below, you just see the natural mineralization of soil organic matter. No cover crop, no fertilizer on that field. So you can see everything's following the same trend, which is likely due to the amount of moisture that's in that soil to help those microbes break it down. So if you've had an active rain event, microbes might be more active and more likely to break it down. So in this case, you can see that after about 4 weeks is where we see the biggest separation between the different tillage techniques, termination techniques, with disking being the fastest, followed by flailing, spraying, rolling. But the disking is pretty far separated from these, but the methods that aren't incorporating are pretty much the same. And after 6 weeks, you see the highest amount of nitrogen. That's when the peak nitrogen release is taking place. But by that point, all the methods are the same. So there's no real difference in when that nitrogen is being released. But however, if you look at hairy vetch, we see some bigger differences. So in this case, we have same thing as before. You have the nitrogen fertilized treatment, the organic matter. It's already in the soil, mineralizing and releasing nitrogen, compared to these different treatments. So after 4 weeks after kill, you see that this incorporation method is really what's releasing the most nitrogen at that point. The other three are about the same. And after 6 weeks, we see more of a separation, where rolling, in this case, has then surpassed disking, not by much, a little bit, and you see some great separation between the disking, flailing, and then the spraying. So we need to look a little bit more closely at this, but it does look like, due to the position of that residue in the soil or on the soil surface, there's either more or less nitrogen being released at any point in time, because the microbes have been sitting on the surface in a mulch, as was pointed out. Those microbes aren't going to have access to all of that material. They can only start decomposing that thin layer on bottom that's in contact with the soil. So that's a little bit where we're going, with our research now. I'm going to skip this in the interest of time. The last thing I want to mention today is a little bit about why we should be using legume cover crops. And one of the biggest arguments we have for using them is that they contain a nice amount of nitrogen with lower amounts of phosphorus when compared to other options that we have for fertility management, such as manures. So if we look at these manures, what we're seeing here is percent of nutrients contained in these different commonly used amendments. And these are from a textbook. These aren't from data that we've created here. But as you can see, there's a lot of nitrogen here, and then if you compare it to the phosphorus that's contained in the manures, you see that these manures also contain a lot of phosphorus. So we really want to think about over-application of manures, because when we're managing for nitrogen and we're trying to increase the nitrogen levels that are available to our corn, yes, we get a lot of nitrogen from these inexpensive, abundant nutrient sources, but we also get a lot of phosphorus. So if we're entirely dependent upon manures, we're going to have a problem over time with increased phosphorus levels in our soil, which can be bad for a number of environmental reasons, due to runoff, eutrophication of waterways, etc. Whereas if we're managing our legumes correctly, we can also get a fairly high nitrogen concentration, but then we don't get that phosphorus loading that we would get with a manure. So my approach is always to think about balancing, using your legumes to the best, the most efficient and best way possible, and then filling in the gaps with manure. One resource that you should all be aware of if you're interested in managing your legumes better, or cover crops in general, is this publication that's put out by SARE, Sustainable Agricultural Research and Education. It's a free publication online. You can buy it. I think it's like $20 if you want a hard copy. But this is something that can walk you through some of what I talked about today, some hands-on techniques you can use in the field for managing your legumes, and basically making you more informed, better managers, so that you can start to do your own experimentation on your farm with some of these things I've talked about today. So with that, I will stop so we can give Chris some time, and probably can take a couple questions and then let Chris take the floor. Do we have any questions? Yeah.
What could be the importance of the investigating working
the advantages of rotating different types of legume cover crops. Well, in general, crop rotation, you're going to have different rooting dynamics, and you're going to be able to take out some of the residual nitrogen that's in the soil by having different architectures able to reach different points. But in terms of legumes, we found, I did present some of this, that if you bring in different legume species, you're going to introduce a variety of Rhizobia types into that soil, which then might stick around for later plantings of other cover crops so that you might not need to inoculate on such a regular basis. But that's one advantage. But in general, having a variety of cover crops in the fields will also increase the nitrogen you're likely to fix and may reduce your risk. So things like that are true across the board for rotation. Yeah. There's actually one other thing I should mention. When you're thinking about planting mixtures, so you have a grass, say, with a legume, remember the principle I put up of the legume fixing less nitrogen or more nitrogen when soil levels are low? If you have a grass that's planted in a field next to a legume, that grass is going to take up soil nitrogen because it can't fix nitrogen. That soil nitrogen is then going to go down, it's going to be reduced, and the legume has been found will fix more nitrogen when it's in association with the grass crop. So that's another thing that you can think about when you're planting grass and legumes, is you can, in some cases, encourage your legume to fix more nitrogen when it's planted with the grass, because the grass is effectively reducing the available soil nitrogen pool.
When we're figuring out nitrogen contribution of a cover crop, I'm wondering if you go out and bail that cover crop, are you taking all your nitrogen, or do you give yourself some credit for the root mass?
Yeah, there's not a lot of nitrogen in the roots. Most of it's, when you think of nitrogen, you can think of it as being green. So if you're taking it away, yeah, you are taking away most of the nitrogen. There's a little bit there, and there's some in the nodule, to be honest. Yeah, I said it's not leaking out. That's not where most of it comes from, but there's still some there. So you would get a little bit, but not as most of it. Yeah, we haven't. It's anywhere from 10 to 20% maybe is left, but usually it's on the lower end. Yeah. So one last question.
I'm curious about, I know one grower in South Carolina that does an understory area to keep crops in his grain with clover, and so then that speaks to what you just said.
So intercrops is grain clover, and clover is an arrow. Yeah.
It's wheat is an understory of clover, so I'm wondering if you're then getting out with
Yeah, it depends. So the question is if you're intercropping. Yeah, if you're intercropping a small grain with a legume. I mean, my guess would be yes, but I don't, it's on a case-by-case basis. You need to look at the system. And there's, if he's tilling in that cover crop later, yes, that nitrogen will become available, and there's still a little bit that's being exuded from the roots during growth, but most of it's going to come from killing that cover crop after, when you need the nitrogen.
I wonder about the economics behind that too, because if you're doing that and it does fix more, then, it's going to go up to the next crop anyway, right? Like it's the big picture.
Yeah.
It's not just this is the cost of this particular crop produced.
Right. And maybe what I'll do is I'll show this slide that I skipped before when we're thinking about the equivalent price per pound of nitrogen. It's going to entirely, the bottom line of this is that it's going to depend on how much nitrogen you produce. The more you produce in that cover crop, the lower you're paying per pound of nitrogen for that cover crop. Cover crop, this is Carl Crozier's data, who's in our department, soil science department at NC State. It's about $60 an acre for crimson clover, $75 for hairy vetch. A lot of that is in the seed cost. And as you produce more nitrogen, so in this case, say you get 150 pounds of nitrogen, you're paying less than $1 per pound of nitrogen. So yeah, if you increase your nitrogen content, your price goes down, logically. So I think I'll stop there, and so Chris has some time to talk, but I'm happy to answer questions later as well. So thank you.